Hi everyone, I'm Lynnea Olivarez, Chair of ASGCT's Communications Committee, and you're listening to Soundbites of the annual meeting, our mini podcast series recorded live here at ASGCT 2025 in New Orleans. Joining me now is Dr. Stephen Kaminsky, Professor of Research in Genetic Medicine at Weill Cornell Medical College and lead author on a Phase 1 clinical trial of a new vaccine for cocaine addiction. Dr. Kaminsky, welcome.
Thank you.
Let's start with the basics. How does a vaccine for cocaine addiction work, and what makes this approach different from other potential treatments?
So the concept is very simple. An antibody bound to the cocaine molecule will prevent it from crossing the blood-brain barrier and not get to its receptors in the brain, and therefore there'll be no reward mechanism from taking cocaine in the first place. The challenge, though, is to make an antibody that recognizes a small molecule, which is typically not immunogenic. And so what we've done is we've taken a compound that looks like cocaine and we've coupled it to a very immunogenic substance that's the adenovirus capsid protein. So adenovirus is typically used for gene therapy, but it's known to be wildly immunogenic. And what we've done is we've dismantled the adenovirus capsid proteins and coupled a analog to cocaine molecule, and then we've formulated it with an adjuvant and delivered it into both mice, rats, and large animals, and shown that it's efficacious in terms of protecting against the normal phenotypes of cocaine addiction.
So fascinating. And this was a blinded placebo-controlled study in chronic users. And what have you seen so far in terms of safety and immune response? And you were telling me just before we recorded this that you just unblinded the data, so
right.
It's super exciting.
It's only been unblinded a couple of weeks. And so what we now have seen is that it's basically safe. I mean, there's some typical adverse events you see with any vaccine, soreness at the site, a little bit of nausea, those kinds of things, typically seen in many vaccines. The only adverse event that we saw that seemed out of the ordinary was actually in the placebo group. So obviously it was not related to the vaccine, and it was something else that particular person had. So the safety profile was excellent. What we saw is that we saw very high titer antibody responses to cocaine in all three cohorts. So there were 3 dose cohorts, 100, 316, and 1,000 micrograms delivered intramuscular. And the same titer, you basically peaked regardless of the dose of the vaccine given. That titer was associated with high affinity antibody response binding to cocaine. And we also saw some measures of efficacy in terms of cocaine use and craving.
And backing up a little bit, who was eligible to participate in the trial? And thinking bigger picture, how might that inform how the vaccine could eventually be used in real-world settings?
So we looked at people who were severely addicted to cocaine. That's actually an official category in the addiction world. And we basically randomly signed them to either placebo or vaccine groups. And how it plays out in terms of a business model and how it plays out in the community of addiction is not totally clear at this point, but it would probably work for people who are addicted that want to get off. So they would have to volunteer to basically accept the vaccine. There's a very large number of people who are perpetually addicted to cocaine that are really looking to stop.
Yeah.
So I think that's the population that we would go for.
Yeah. And finally, could this type of vaccine platform be applied to other substance use disorders down the line?
Yes, so we've actually published on this vaccine concept for nicotine, and it worked quite well. We published on a slightly different concept where we encapsulated a nicotine antibody that we knew was high affinity into an AAV particle and delivered that, and shown that the expression of this antibody specific to nicotine worked well in a mouse model as well. The advantage of that concept would be that you don't have to depend on the genetic background of the recipient, so you're delivering to them what you know is a high affinity antibody. We are currently working on a vaccine for fentanyl, at the same concept with the disrupted adenovirus, and that would be the protection against overdose.
Wow. This is as real world as it gets. Super exciting, Dr. Kaminsky. Thank you so much for joining us, and we'll certainly be keeping a close eye on what's next for this important program.
Thank you.
So for all our listeners, stay tuned for more sound bites of the annual meeting.